Hello, 3-Day community. Happy New Year. We are so excited to have you guys back with us for another 3-day webinar as we, continue our series, highlighting our different researchers across the country that benefit from Komen funding. We have officially hit 2017, which means we are in full gear for the 2017 season that will kick off in August. Hopefully you guys have registered, are recruiting, getting the information out there so we can have a really successful season. But with that, I'm going to kick it off to Chrissa Smith, who will introduce our scholar and take it away.
Hi, welcome to today's webinar. I'm Chrissa Smith, the Senior Manager of Scientific Review. I'm over the application process where we give research awards to scientists. And I am delighted to introduce today's Komen researcher, Dr. Jay Desgrosellier. For those of you who walked in San Diego, he was at the camp show. His collaborator and mentor is Dr. Jeff Wall, one of our Komen scholars, and who spoke at the camp show in San Diego. I'm also based out of San Diego, and I've had the opportunity to participate by cheering and volunteering at the 3-Day for years. I've worked for Komen for about 5 years, so I have a lot of pink in my life, but I have to say the 3-Day community puts me to shame. I'm incredibly impressed with the spirit and passion that they bring to San Diego, so thank you for bringing that. One of the things we've learned over years of breast cancer research is that the problem is incredibly complicated. We really need smart, passionate, and dedicated people to address that problem and take on that challenge. And Dr. Desgrosellier, an assistant professor from UCSD, is one of those people. He's taking the question back to the very basic question with a research grant from Komen. He's looking at early cell changes that give the cells specific growth characteristics that can lead to aggressive tumors or metastasis or recurrence. His goal is to target those few specific cells that lead to poor outcomes instead of just targeting the main bulk of the tumor. So description of basic science can be a little bit intimidating, but luckily he's here to walk us through some of the exciting concepts of how these stem cells may be the key to treating metastasis. So thank you for joining us today, Dr. Desgrosellier.
Thank you. All right. Well, hello, everybody. It's good to be with you. As Chrissa mentioned, my name is Jay Desgrosellier, and I'm an assistant professor here at the Moores Cancer Center at the University of California, San Diego. And today I'll be discussing a little bit about how Komen research dollars are at work to combat metastasis. Why aren't we advancing? There we go. All right, so I thought I'd start by sharing a little bit about my background and why it is that I study breast cancer. I think my journey is quite similar to most scientists, where we usually start out studying something completely different from where we end up. And so I actually began by studying developmental biology and cardiovascular birth defects in graduate school. And I received my PhD degree in pharmacology from Vanderbilt University. Now, pharmacology is not to be confused with pharmacy. Pharmacology, very broadly, is just a study of how drugs affect the body. So I really didn't even study cancer until my postdoctoral fellowship. And it's during this time that I really developed an interest in solving big problems in science. Now, scientists often get bogged down in some of the minutiae, some of the details, but I decided I didn't want to do this. I wanted to study the big problems that made a difference in people's lives. So as an early principal investigator, or PI, I decided to study breast cancer. I saw in the breast cancer field that there were still many big challenges to be faced and a need for fresh new ideas to address these challenges. I thought that my previous background in developmental biology and pharmacology might give me a new perspective that hopefully could lead to breakthrough approaches in the fight against breast cancer. So Komen funding is absolutely essential for early career scientists like myself. So here's a picture of me in my laboratory. So I'm shown here on the right. And this is Elaine here on the left. And Elaine is an undergraduate here at UCSD who's been volunteering in my lab for 1.5 years now. And I also have one other volunteer now in the lab, Daniel, who started with me last October. And so you can see that I have a pretty small laboratory. And so Komen funding through the Career Catalyst mechanism has really been essential to me having an academic career in science. Additionally, Komen funding is really important for early career scientists like myself to establish our independent research programs. So as a postdoc, we often work under a senior mentor, and it can be really difficult sometimes to establish your own independent niche. And this is absolutely essential in order for us to compete for the stable, long-term funding from the National Institutes of Health, or NIH. And these grants are really important in order to build a laboratory, not only to justify space in the institution, such as the lab bay you see here, but also to recruit the people who actually do the work in the laboratory. So as I advance in my career, I find that more and more I'm tied to the computer, and I spend less time in the lab. And it's really the folks like Elaine here who are out there in the lab doing the experiments that are furthering the science. And so speaking of the people in the lab, Komen funding is absolutely essential to training the next generation of scientists. And Elaine here is a good example of this. So as I mentioned before, Elaine's an undergrad here at UCSD. And she's actually in a program here where once she graduates, she's already admitted to medical school. And so Elaine just found out she's actually going to be graduating at the end of next quarter, so as an 18-year-old sophomore. So obviously she's a very exceptional student. And now Elaine is beginning to express an interest in doing breast cancer research after her medical degree. And so this is exactly the kind of feed-forward effect that Komen funding can have. Not only helping people like me to build our laboratories, but also recruiting and training the next generation of breast cancer researchers. So before I get into my own research, I first wanted to touch on some of the progress that's been made in the breast cancer field in the last few decades. I think chief among these achievements have been is the fact that radical bilateral mastectomy is no longer routine. So I remember my mom telling me about when she was 21 years old, she went in with a lump in her breast to get a biopsy. And the doctors put her on their operating table and told her that if it's cancer, they're going to remove both of her breasts. Now, fortunately, it wasn't cancer, but I can't believe anyone of any age having to go through such an experience. And so the fact that this is no longer a routine procedure is a major accomplishment. Additionally, we now have targeted therapies to treat specific types of breast cancer. So for example, if your tumor has the estrogen receptor, then we'll treat you with anti-estrogen therapy. Whereas if you have a cancer that overexpresses the HER2 receptor, then we'll treat you with this monoclonal antibody called Herceptin, or trastuzumab. And both of these are highly effective therapies at treating their specific types of cancer. Additionally, we now live in the age of genomic testing, where doctors can take a piece of your tumor and sequence the DNA and actually determine the specific genetic mutations that are driving that cancer. We can then use what we call precision medicine, which is basically where we take a drug off the shelf that will specifically target your individual mutation based on your own personal tumor genetic profile. And so this is a major achievement in the field, and it is now basically the state of the art in regards to breast cancer treatment. And let's not forget that surgery, chemotherapy, and radiation actually have worked to help many women survive breast cancer. And so there have been many big achievements in the field in the past several years. But there's still a lot of work to be done. I think we can all agree that there are far too many deaths from breast cancer, with more than 40,000 each year. So I recently noticed that Komen has a new goal to reduce the current number of breast cancer deaths in the United States by 50% by the year 2026. And I think this goal is to be applauded. It is exactly the right kind of goal for an organization like Komen to have. So if we're going to talk about reducing deaths from breast cancer, we have to talk about metastasis. Essentially, all breast cancer-related deaths are due to metastatic disease. So if we're going to prevent deaths from breast cancer, we need to ask, how do we treat metastatic disease? And I believe that this is exactly the right question for us to be asking at this time. So my laboratory focuses on metastasis. So what is metastasis? Well, quite simply, it is the spread of tumor cells to distant organs. So literally, these cells get up and move from the breast to distant organ sites. So if you have a primary breast cancer, like the one shown in the schematic here by these blue cells, some of these cells may move to distant sites such as the lung, the bone marrow, or even the brain, where if they happen to grow, they can cause organ malfunction, which can lead to death. And so we now know that moving is actually the very easy part for these cells, and that most of the cells that get to these sites aren't capable of forming metastases. So why is this? Well, tumor cells actually have a great deal of difficulty growing in other organs, and this is a concept known as dormancy. So I like to use this picture to illustrate this concept. First of all, because it has birds in it, and I like birds. But second of all, I think one can imagine that if this adult penguin here is like a breast cancer cell, and it happens to end up in a place like the lung, and so all these baby penguins maybe represent all the lung cells, you can see just how out of place these breast cancer cells are when they arrive at one of these distant sites, and just what kind of an uphill battle they face in trying to grow here and establish a metastasis. And so I think the lesson to be learned here is that we don't need to target every cell that reaches these distant sites, just the ones that are actually capable of growing and forming metastases once they get there. And so how do we identify the right cells to target? Aren't all breast cancer cells really just the same? Well, actually, tumors are quite complex. So while we often view cancers as one big amorphous blob, kind of like Homer Simpson here, with maybe a few different colors, such as a green one, a blue one, a red one, we treat them as all basically just the same. But actually, within a single tumor, there may be many different types of cells. And I think this puzzle here kind of illustrates this concept nicely, showing all the different colored pieces making up a puzzle, much like there are many different cells that make up a primary tumor. So within a single breast cancer, there may be cells that harbor different genetic mutations. Just like this red puzzle piece here may represent a cell with one genetic mutation, whereas this green piece may represent a cell with an entirely different mutation. And just like this puzzle is made up of different colored pieces of different shapes and sizes, similarly, cells within a given cancer may look and act differently. You may have cells that are elongate and very migratory, and you may have cells that are very cuboidal and very proliferative. Additionally, there may be a lot of similarity with normal mammary cell types within a given breast cancer. So actually, there's quite a bit of diversity in the normal mammary gland among the different cell types, and this diversity may be reflected in some breast cancers as well. So most interesting to us is the fact that there may be cells that are similar to normal adult mammary stem cells within some breast cancers. And so maybe it's these stem-like tumor cells that are actually the right cells to target to prevent metastasis. So the approach we use in my laboratory is to ask, what can these mammary stem cells teach us about metastasis? So work from my lab and others has shown that there's a population of adult mammary stem cells in the breasts of women that are important for the remodeling processes that occur during pregnancy that allow the gland to secrete milk and feed the infant. Now, it's important to point out here that these are not embryonic stem cells. These cells occur only in adult women and only in the breasts. So what is a mammary stem cell? Well, we define these cells quite simply. Mammary stem cells are capable of forming a mammary gland when injected into an empty mammary fat pad, as shown here in white. And so these cells will reconstitute a glandular structure, as shown here in blue. So what does this look like in real life? Well, here's an example of a fat pad where there are no stem cells, so there's no gland present. And if stem cells are present, then you actually get something like this, where there's an outgrowth consisting of many ducts and alveoli, and this is stained in red here so you can see it. And it's this ability of stem cells to form an outgrowth that makes them likely candidates to contribute to metastasis. So my lab asked a very simple question. Do these stem-like cells exist in breast cancers? And so I'm showing you right here some exciting new data from my lab where you're actually seeing these cells for the very first time. So this is an actual patient tumor. This person happened to have a tumor that was HER2 overexpressing and lacked the estrogen receptor. And in this black box here, I'm showing you an example of one of these stem-like cells that is stained with two different markers so we can see them. And these are the cells that we believe are like the stem cells I just showed you in the previous slide and are capable of forming an outgrowth. So we identify these cells with two different markers. One stains the nucleus brown, and the other stains the cell membrane in blue. And it's the presence of both of these markers that make them stem-like cells. And you can see this cell here at higher magnification. Excuse me. So these cells are quite rare, and most of these cells lack both markers, but there are a few cells here that contain one of the markers. For example, these cells have brown nuclei, but no blue. So these would not be stem-like cells. So our ability to identify these cells in actual patient tumors now allows us to ask how these cells might contribute to metastasis. So we hypothesize that these stem-like cells may act much like a queen ant within an ant colony. So for those of you who know about ants, you know that the queen ant's the only one capable of reproduction. And you can kill off as many of these worker ants as you want, but you'll never kill off the colony unless you get rid of the queen. So much like a queen ant, we think these stem-like cells, if they happen to travel to some place like the lung, that they'll actually be able to form a colony or form a metastasis. And just like an ant colony, if you use a therapy like chemotherapy that gets rid of the bulk tumor cells, just like getting rid of all the worker ants, it leaves behind these stem-like cells, which may act much like a queen ant and replenish the colony. And this is what we call a recurrence. And so this is how we hypothesize these stem-like cells may contribute to metastasis and recurrent disease. So what's in store for the future? Well, I think much like we have precision therapy now based on genetics, in the future we'll have precision therapy based on the cellular composition of the tumor. So for example, if you happen to have these stem-like cells, we'll use a specific therapy that targets that particular cell type. Whereas if you have a different aggressive cell type, we'll use a different type of therapy. Now, my lab has recently discovered a specific drug that can kill off these stem-like cells I just showed you. And we're really excited about this drug and the possibility this may provide a new way of treating breast cancer. So this is how we envision this happening. So if you have a primary breast cancer with some of these stem-like cells, which are shown represented here by the red cell, so as I previously showed you, chemotherapy will not touch these stem-like cells, so you'll get rid of just the bulk tumor cells. But if you now add an additional therapy, like the one we've identified, that can kill off the stem-like cells as well, now these cells will no longer survive after chemotherapy, and we can prevent new metastases and recurrences from forming. So where does this lie along the research timeline? So some of you may be familiar with the fact that research progresses from the basic research or discovery phase into clinical trials and finally for end use in patients. And so while we're still here at the basic research or discovery phase and are trying to validate this approach, we're using drugs that we're actually repurposing. So these drugs have already been used in clinical trials for other purposes. And so by using these drugs, we hope to expedite the transition into clinical trials in the very near future. So this is all possible because of you. It's really this simple. This discovery doesn't get made without your support. And Komen support is more important now than ever before, with federal funding becoming scarcer and more competitive than ever. Additionally, private funding sources such as Komen are really important for scientists to take the calculated risks that are necessary to drive bold new discoveries. So federal funding often rewards past research accomplishments and incremental advances. And so it's funding from private foundations like Komen. It's really important for the risk-taking that will spur the new discoveries that will really make a difference in people's lives. And this is incredibly important for early-stage investigators like myself. We need to be able to make these bold new discoveries in order to justify institutional investment and federal funding in the future that is necessary for us to really build our laboratories. And finally, Komen makes available to researchers like me, science managers like Krista here, who greatly aid in the research progress. And Krista has already been very helpful to me by introducing me to a collaborator at the University of Utah who's really been a tremendous help in furthering my research. And so finally, I just want to thank all of you and thank all of my funding sources, in particular Komen, and especially the 3-day ENRACE participants and their donors, friends, and family. It is really through your guys' efforts that we continue to make progress in the fight against breast cancer. You guys let me do what I do, and I hope that together we can help Komen in their goal to reduce deaths from breast cancer. So please keep it up. Thank you.
All right, well, thank you, Dr. Desgrosellier. So this is I'm David, also a Komen Research Grants Manager that's actually stationed in Nashville, Tennessee. And so if you have any questions, feel free to ask those questions via the question box or comment box. I see a couple of them that are starting to come in. And so I guess one of the first questions I have for you is concerning we have come a long way, but I'm curious as to what you think the next big step might be in reducing these breast cancer deaths. What do you see as being the sooner step that could be taken that could lead to the greatest effect?
Well, first, I think my presentation, I was trying to make the point that it's identifying the right cells to target that's really the most important piece of attacking metastasis. Obviously, we've been trying for years to treat it with chemotherapy, radiation, all the things that we target the primary tumors with, but it just doesn't seem to have any effect. And so I think we need to take a step back and take a very new look at how we treat metastatic disease and how it might be different. And I think the answer is that we're just going to need to identify these appropriate cell types to target, and they might require some different drugs that we already have available, like the one we've identified, like the SRC inhibitor, dasatinib, that we're using. It's already been tested in clinical trials and used on other diseases. It's just a matter of finding the right cells and the right targets within those cells in order to prevent their role in metastasis.
So another question would be, with some of these repurposing of some of these drugs, do you think that they're going to be targeting the same kind of pathways, signaling pathways, or do you think there could be other ways that can make these repurposing these drugs target new pathways potentially?
Well, I think they're going to be used to target new pathways. Once we identify the correct cells, we're going to discover that maybe they're using some of the same molecules we're familiar with, but using them in a different way. And so some of the drugs that we already have on the shelf may be very effective at targeting those cells if we just use them in the right way. So the other thing I was trying to point out in my presentation here is that we need to treat breast cancer differently, especially metastasis, if we're going to have any success. Just about everything we've done so far has been an attempt to reverse pre-existing metastases. So once the problem's already there and so at a critical stage, we're trying to get rid of it. And it might be a much more effective strategy, since most metastases don't even occur until later in disease progression, after the person's primary tumor has been removed or in remission even. If we can somehow treat with the drug at that point, before the metastases become established, before they've really started to grow and cause problems, maybe we give the appropriate drugs that kill these cells, like the one we're using, and it might prevent the metastases from ever getting started in the first place. And so we might have to rethink entirely the strategy of how we give these drugs and the context in which we give these drugs.
And I guess a follow-up question to that, do you see potentially the targeting of this more along the lines of allowing the stem cell to, or the stem-like cell to grow out at this secondary site, this metastatic site, or do you see potentially preventing it from, getting to that point? So do you think it's going to be preventing it en route, or is it going to be preventing it from, growing out?
I think the most effective means of preventing metastasis will be to prevent those cells from even ever getting to those sites. Because once they do, as I was kind of illustrating in my schematics, they give rise to a diversity of other cell types that will no longer be affected by the same therapeutic strategies, same drugs. And so it'll be much easier if we can treat the stem-like cells, before they really get established in what they call the adjuvant setting, so post-chemotherapy or post-surgery. That'll be the time to use these drugs, and they won't have to be used forever because they actually kill the cells. So that may be a good use for some of these targeted therapies in the adjuvant setting to prevent these stem-like cells from becoming established and growing new disease, new metastases and recurrences.
Great. If there are any other questions, feel free to ask these questions via the question box or chat box. And so I'm trying to see some of these questions that are coming in. So I guess another question here that I see is revolving pregnancy and lactation. And you mentioned something about these adult stem cells. Can you expand a little bit more on the pregnancy lactation and how the adult stem cells have a role in that?
Oh, sure. So that's essentially why the stem cells are there, why they exist. The particular stem cells we're talking about are entirely hormonally driven. Excuse me. So they've done experiments in mice where you actually get rid of the hormones and the stem cells go away entirely. So you have to have estrogen, you have to have progesterone, the steroid hormones present to actually drive the stem cell behavior. And so obviously during pregnancy, what do you have? You have more hormonal signaling, and that drives an increase in the number of these stem cells and in the ability of these stem cells to give rise to other cell types, including the cells that will go on to secrete milk during lactation. There's a huge increase in cell number and remodeling of the mammary gland that has to occur during pregnancy in order for it to be able to produce milk and lactate. And so it's the stem cells that are present that respond to the hormones that actually are driving that remodeling process in order for lactation to occur.
Which I guess a good follow-up with that too is, does hormonal therapy potentially impact these adult stem-like cells and these adult stem cells since they are hormonally driven?
The normal stem cells are regulated hormone, and that might occur. We don't know this yet. That's an excellent question in regards to cancer. We do know that there are some of those cells that are stem-like that escape the hormonal control. The hormonal control is indirect. The stem-like cells actually don't have hormone receptors. They are receiving messages from other cells that do, and so it's indirect. And some of these cells we know escape that regulation. But it's entirely possible that in some cancers they retain that hormonal dependence, and that's a very exciting thing for us to follow up on and to examine now that we can identify those cells in tumors.
Awesome. Well, I think that's all the time that we had allowed for the 3-day webinar. So on behalf of the 3-day group, I'd like to thank Dr. Desgrosellier for speaking to us and doing this webinar. If you have any other questions, feel free to submit those questions. I think there's a link that we're following up with the GoToWebinar, and so you can send those, and we will send those questions on to Dr. Desgrosellier to submit those questions for his answering, and we'll get those back to all the attendees of this 3-day webinar. So thank you all for being a part of this. Like I said, this webinar will be available for viewing down the road, and so we will get that sent to everybody following the close of this webinar. So thank you all again, and have a great rest of your week.
All right, thank you.
Thank you guys so much.